Good morning and good afternoon, everyone. My name is Harris Juvali. I'm part of the global marketing team at Olink, and I will be your moderator for today. Welcome to this month's edition of the Olink Multiomics Coffee Break Conversations. Together with our customers, we aspire to reveal the truth of human disease protein by protein. This is a series of webinars designed to show you what is possible to do with proteomics as an additional layer on existing omics data or as part of your ongoing multiomics projects, and hopefully inspire you to start thinking of the possibilities that next generation proteomics opens up for your research. Today's webinar is organized in collaboration with AGRF, an Olink certified service provider. AGRF is part of an Olink certified service provider network, which consists of a great number of CROs and core labs that are ready to support your projects for proteomics and beyond. You can outsource your samples with confidence and trust them to deliver top quality data to accelerate your science and support you with your multiomics integration. Today we have with us Desley Pitcher, Head of Sales at AGRF, who will do a brief introduction to AGRF, and we will continue with Dr. Tom Mincherton, Research Officer at the Children's Cancer Institute in Australia, who will help us dive into advancing pediatric cancer monitoring through NGS-based proteomics. We also have with us Satish Bhaskaran, part of our support team here at Olink, to help us with any technical questions you might have. And with that, Desley, the floor is yours. Thank you.
Thank you, Harris. It's lovely to be here, and thanks so much for having us. Hello, everyone. My name's Desley. As Harris said, I'm the head of sales here at AGRF. We're starting as a genomics organization who has taken on proteomics, and we selected Olink to be our proteomics supplier. And we've been running for well over a year now, and it's very exciting technology for us. So starting with a little bit about us, we are Australia's largest genomics service provider, and we've been around for more than, actually that's 27 years now, delivering genomics excellence to the Australian community, but also beyond. We are an independent, not-for-profit company, and we do have a national network of scientists around the country in Adelaide, Brisbane, Melbourne, Sydney, and Perth. And those labs support all of our clients across the country and, as I said, internationally as well. We do have NIDA accreditation to ISO 15189 for clinical testing, as well as standard ISO 17025 for biological testing. So we're covering the human pathology, animal health, and life sciences. We pride ourselves on having a track record of early adoption of new technologies, and through that, we've developed a strong national collaborative network with researchers, commercial organizations, as well as diagnostics. So just a little bit about our people. I've already mentioned the cities that we're located in, and we're co-located in very strategic places. So we're supporting people doing really impactful research in each of those cities. We have well over 110 highly skilled staff and support more than 450 research and commercial organizations every year. And it's well over 4,000 individual clients each year that we're servicing as well. We do have a dedicated innovation and development team, which I'll talk about a little bit more in the next slide. As I said, well over 3,000 or 4,000 clients and collaborators that we're working with. A very broad overview of our comprehensive suite of services. So our biggest technology is next generation sequencing, and that's one component that's drawn us along to Olink, which is our non-mass spec-based proteomics. We have solid long-read sequencing platforms as well. We offer standard Sanger sequencing, genotyping, and particularly with array genotyping, but also custom SNP. And we do have very strong support services in our nucleic acid extraction technologies, as well as a big bioinformatics team. And as I mentioned, an R&D team who is responsible for developing new technologies, building out those technologies and being able to bring them to service. I did also want to briefly touch on those accreditations that we have, particularly in the clinical space. And this is where we'll be supporting CCI further into the next half of 2025. So we do have 15189 accreditation, as I mentioned, which allows us to deliver secondary analysis through to pathology providers. We have a standalone quality management system and a dedicated quality and regulatory affairs manager, and we do participate very regularly in quality assurance programs in order to maintain our high accreditation status. I've also listed out the services that are specifically 15189 accredited here at AGRF, but I won't bore you with reading through all of those. So just stepping into the Olink Proteomics platform, we decided to bring on protein biomarker discovery with Olink because we could see a really strong link between genomics and proteomics. Of course, multi-omics solutions are looking to be the way forward. The applications that we sit in are targeted biomarker validation, very large-scale discovery projects, particularly in population studies, and we've got quite a few of those happening at the moment. Of course, disease research, we have some in drug discovery and development as well, and of course, we're able to give really comprehensive data insights, especially if you combine that with some of our genomics studies as well. We started with the Explore HT system, and we've also now added Olink Target in partnership with Telethon Kids Institute in Perth, and of course Olink's newest technology, Reveal, as well, as one of our suite of services. So my next slide is the last one that I wanted to show you today, and it's just wanting to outline a few of our client projects that we have been supporting since we kicked off our service, I think in April 2024. Some really exciting ones in the space of spinal cord injury, colorectal cancer and other type of cancers, a very large population study on an autoimmune disease. We've worked with the Kids Institute on cystic fibrosis, and we're just starting 2 individual drug treatment trials looking at longitudinal response. So it's a really exciting area for us. We're very keen to see how some of these reports come out. We're interested to see these results, and we're very proud to support, in particular, these projects, but also supporting CCI as well. So I will end there and say thank you very much, and hope I can introduce Dr. Tom Mincherton to continue on with the presentation. Over to you, Tom.
Hello everyone, and thanks for having me here. I am Tom from the Children's Cancer Institute in Sydney, and again I'll be talking to you about advancing paediatric cancer monitoring through next generation sequence-based proteomics. So to start, we work in childhood cancer, and this is a global issue. There is over 400,000 cases diagnosed every year. Here in Australia, there's about 1,000 children every year diagnosed with cancer, and it kills approximately 3 children every week, which results in a massive loss of life and potential. These kids on average have a 70-year less life than they would have if they hadn't had cancer. Even those that are successfully treated, over 70% of them will have long-term effects from their treatment. And we really want to work on improving this for all the children that are out there. The treatment of childhood cancer has 4 key pillars, which are surgery, chemotherapy, radiotherapy, and immunotherapy. There's a lot of work going into improving all of these, but in particular, immunotherapy still lags behind the results that we see for adults with cancer. And we think a lot of this is to do with the fact that children have a different immune system to adults, and there needs to be a lot more work going into understanding this so we can improve their results. That's where it brings us to the Zero program launched out of CCI, which aims to improve the results for all children who are diagnosed with cancer. How the Zero program sits into the average journey of a child who is diagnosed is after they get sick and go to hospital and they are diagnosed with their cancer, a sample of that will be sent to the Children's Cancer Institute, where we will do an array of techniques, including whole genome sequencing, RNA transcriptome sequencing, methylation profiling, and liquid biopsy monitoring, which is where the work I'm speaking to you today fits into this story. But all of this is brought together and compiled and shared with the child's doctor to try and refine the treatment that they are undergoing and giving them a better chance of survival. There's also further testing conducted, especially in high-risk children where possible, including in vitro and in vivo drug screening, to try and again correlate with their doctors and refine that treatment even further, all to provide better outcomes with children with cancer. The currently operating 02 program has enrolled over 1,600 children, and this is across a wide range of different cancers from leukemias, lymphomas, central nervous system cancers, sarcomas, the works. Over the entire 0 program, there's been over 2,700 children enrolled. And what we want to do is use this massive amounts of samples at our disposal to really understand the children's cancer environment better and how we can improve their treatment. And as I said, one of the key interests of VIRA is the immune system and how it can be used to correlate with their immunotherapies. So one of the things that was developed at CCI is an immune pediatric signature score developed by Chelsea Mayo and her team here. And what this does is use gold standard immunohistochemistry combined with a lot of machine learning and AI, and developed a 15 gene signature, which can be used to define cancers as either hot or inflamed or cold, where there's not much immune cell infiltration. And what these genes do is allow us to define the level of infiltration within the cancer. If you want to learn more about this, I would direct you to Chelsea's wonderful paper that's on it. But this is really the forefront or the starting point of our look into the immune system. But we wanted to take this further again, and this brings us to the ASPIRE program. And what this wants to do is do high throughput, high dimensional, spatially resolved analysis that is compatible with the clinical samples we already have available to us. So what this integrates is both spatial transcriptomics and proteomics, as well as investigations into peripheral immune system, looking at peripheral immune profiling, as well as plasma proteomics with the Olink system. And we want to bring all of this together into an easily accessible and analyzable data sharing portal for the clinicians in the future. But to bring the proteomics into this, one thing that is important is that it is compatible with the samples we already have available. And we collect a lot of our peripheral bloods in either STREC or EDTA tubes. So Wen Young in our team has done a lot of work into making sure that both of these samples are going to be compatible with the analysis we perform. And he's noted that there's no key or differences between the results we get, whether that sample was collected in EDTA or STREC, or what sample type we have collected, which means on everything we've got available, we can proceed with a lot of this Olink analysis. Then he's gone on to show that what we can do is delineate key sample types using some of the plasma proteomics that are available. So for example, we can easily identify CNS tumors based on their GFAP expression or neuroblastomas based on ALK expression. And pushing this further, we can even identify and cluster key cancer types solely by their plasma proteomic profiles. And you can see here at the top, the clustering of our sarcoma patients, and at the bottom, those with central nervous system cancers. However, you don't need the 5,000 proteins we've used just to tell you what type of cancer is. So we want to push this further and start asking even more specific questions. And that's where some of the work done by Erin Cole and our team comes in, seeing what we can tell about the peripheral immune system using the plasma proteomics. And what she's done is take the patients defined as inflamed or cold. This is a pan-cancer approach to see if there's any key signals that are upregulated in either of these two groups. However, this didn't reveal the number of key markers of immune system surface proteins or immune checkpoint proteins that we expected to see in these two groups. And we believe that part of that is because there is so many signals going on and so much just natural noise as a result. It's actually blocking or sort of hiding a lot of those key signals that we're looking for. But what we can do is ask even more specific questions to break this down and start revealing some of those signals of interest. So for example, Erin took this further and looked into cytokine trafficking signals, which are essential for migrating those immune cells from the lymphatics through and into the cancer. And what we saw here is in these inflamed or hot cancers, massive upregulation of a lot of key cytokine trafficking signals in comparison to our cold tumors where we didn't see these same signals. We can even break this down further and start asking disease-specific questions. So in this instance, what Erin has done has taken 4 neuroblastoma cases, all defined as cold by their IPAS signal, and start to investigate if there's any differences between them using the Olink system. And what we could see here is upregulation of a lot of these cytokine trafficking signals in a cold neuroblastoma case. And what this could represent is it beginning to transform into that inflamed state compared to these other ones which had similar IPAS scores but did not have the upregulation of these systems, these signals, sorry. So in the future, what we really want to do is see how far we can push this and ask even more questions. How does this correlate to all of that spatial information we're generating through the Maxima and Xenium systems? Can we use these panels correlating with the IPAS to inform our flow panel designs to get better profiling of the peripheral immune system? Are there signals in this that are evidence of circulating disease, and how does that correlate to cell-free, circulating tumor DNA, as well as circulating tumor cells? Are there signals within this that we can use to identify disease subtypes and define risk categories? While we've done some amazing work so far, we're really just scratching the potential of the Olink system, and there's a lot more yet to do. So I would like to thank my team at CCI, including our executives on the Aspire program, the entire lab group, the extended Zero Network and Computational Biology group. We have a lot of international and national collaborators that have helped with this work, and of course the children and their families who have donated to these studies in such a trying point in their lives. So thank you all for listening.
Thank you so much, Tom. Really, really nice presentation on this very, very important subject of childhood cancer. It really struck a chord when you said 70 years of life lost there. So just before we move to the Q&A session, and please put your questions for Desley and Tom into the Q&A box. I just have a poll, some poll questions for you. Please take the time to answer some of those. Thank you for taking the time. I can see responses coming in. I'll go to the next question. And we really want to make sure that we only contact you when there is an interest, but please be open to just a conversation on your projects and how Olink and AGRF can help you there. And which applications would you like the next webinar to focus on based on your interests? So thank you so much for taking the time to answer these poll questions. And with that, I would like to invite the presenters, and we can start with our Q&A session. So the first question is, what is the actual sample volume that is typically needed? And does that volume change depending on sample type? Maybe Satish?
Thank you, Harris. For Olink Explore HD, we use less than a microliter of sample. But when you send it to AGRF, for example, they might require like 40 to 50 microliter of sample volume. So yeah, somewhere around 40 to 50 microliter, but actual assay less than a microliter that we use. And the second question, the sample volume depends on the sample type? No. So it's the same sample volume, irrelevant of what sample type that you send in.
Thank you so much. And Tom, you described a lot of techniques that you are using to come up with these signatures. Do you find that there is a validation step once you have diagnosed a pediatric cancer? Do you further validate your findings, or do you rely on this signature?
So the diagnosis will come to us through the Zero program. And then what we were doing here was going that, yep, we can identify these key diagnostic markers through the Olink. So when we start pooling our data together and we do these sort of pan-cancer approaches, we can then extrapolate back and go, oh, this signal was more coming from the CNS cancers, or these ones are sarcomas or anything like that.
Okay, amazing. How these signatures come from a certain number of cancers, I suppose. Tom, do you have like a specific signature for each pediatric cancer, so to speak?
Not as of yet, not like a defined this equals this cancer. However, I show you back to this slide. This does give an example of some of the key markers along the bottom here that we can use to do the clustering and identify disease type back when we look through the data. But it's not a set panel. This is exactly what defines this disease as of yet.
Okay. So how many different tumor types have you analyzed is the question. And can you find signatures able to discriminate between all different types of tumors? And can this be used for cancers of unknown origin?
So we haven't tried it on cancers of unknown origin yet. But again, if I go back to this one here, you can see that we've applied it in neuroblastoma, central nervous system cancers, sarcomas, other solid cancers, and vascular lymphatics. And again, we're starting to get an array of genes that can be used to discriminate between these. But what we want to do going forward is to break down these categories even further. So within the CNS, can we define a profile that will separate out glioblastomas from medulloblastomas? Within sarcomas, are there some that will tell us whether it's an osteo or anything else? Yep.
Fantastic. And Tom, this is my question here. Maybe I missed it during your presentation, but are these samples that you're using blood plasma? Or, I mean, my question is, how close are we to a non-invasive diagnosis for children?
Yes, so in the liquid biography program, one of our main focus is being as minimally invasive as possible. So all of this work has been done on blood plasma.
Amazing. Amazing. This is really, really hopeful. All the work that you're doing. Thank you so much. Another question is about the data analysis burden. You described many different omics here, Tom. So how do you manage all this data?
With a massive team is probably the best answer there. So yeah, we do have a lot of different platforms going under the Aspire program. So we've got transcriptomics through the Xenium platform. We have proteomics through, slide-based proteomics through the Maxima system, our plasma proteomics through this, and then of course our flow cytometry data as well. So we do have quite a large team and quite a lot of excellent technical specialists who are experts in their individual fields who tend to manage their portion of it. And then we have a great amount of bioinformaticians and that who are fantastic at bringing all of that back together on the back end. So it is just the fact that we have just a lot of great members, each doing a small part, that allows this to happen.
Fantastic, thank you. And Desley, how can you help someone that is new to proteomics, someone that is in genomics, transcriptomics, any other omics? How can you help support them with maybe, I don't know, study design or exploring possibilities?
Yeah, great question. We do that all the time. We are lucky enough to have a very talented technical officer who works exclusively with our Olink clients. His name is Ben Cooper. I think we saw a photo of him earlier holding a certificate. So Ben takes care of those client interactions. We do have a large team of account managers and clinical genomics specialists as well. So those go out with Ben, they speak to clients and help them to develop their assays. We do rely also on our local Olink support team, which includes Brenton and Satish, who's here on the call today, who also can help out with those designs as well. But we find that Ben gets quite technical with clients and has really strong discussions about study design. One we're working on at the moment is really interesting, one from the US, which is who are doing a clinical trial here in Australia looking at a particular drug interaction and how that might reduce the need for certain types of other drugs in the future for patients, a little kind of in pharmacogenomics space, but we're doing it with protein instead. And yeah, that's been something that Ben has managed alongside that client for about 6 months now, and the samples are about to arrive. So we're quite excited to see how that goes. But yeah, we have a team of people and bioinformaticians as well who help out with that.
Thank you so much. And a question for Tom. Have you overlaid Olink data with other omics data? Did proteomics help to provide better discrimination of different cancers treatment outcomes?
So that one is a not yet. What we want to do is start correlating a lot of this Olink data back to the other proteomics and other investigations we're doing. However, this is a very new project at this point. So we're still in the early stages of validating each of these components individually, and then we can really start bringing them all together. So invite us back in 6 months and I should have an answer.
If I may pop in there, I can talk about a project that we've had where the client has done both whole transcriptome sequencing as well as Olink proteomics. And they found 167, I believe, genes that were upregulated with a particular condition. But when they did the proteomics, they found only 3 proteins that were of interest. So it really narrows down the focus for them on what they need to work on next. That was also with children, and they really appreciated the fact that it was such a small sample volume needed, because as you know, with children, it can be quite difficult to get decent volumes. They were using not plasma, they were using a nasal liquid. So again, very small amounts.
Thank you so much. And this is a good time to ask Satish, actually. We talked about nasal samples, we talked about blood samples, but what other sample types, Satish, can be used with the Olink platform, just so our audience understands?
Thank you, Harris. Olink kit is validated with plasma and serum sample type, but we have experience with the other sample types also, ranging from CSF, urine, or nasal fluids, which are shown to compatible with our method. So if you have a different sample type that you like to produce like Olink data, please contact us, and we have a particular protocol prep that we can provide you with, so that we can run it in a Olink platform. So if you have a technical question on that front, contact us at support@olink.com, and I'm more than happy to support with that. Thank you.
Excellent, thank you. So we have gone through all the questions that we had in the Q&A box. So I would like to thank you all for attending today. And thank you, Desley, and thank you, Tom, very much for presenting today and letting us see a bit of all the exciting work that you are doing. So with that, thank you again and have a good day, everyone.
Thank you.